Hello everybody, my name is Henning Schulze-Bergkamen from the Department of Medical Oncology at the National Center for Tumor Diseases in Heidelberg, Germany. I'm delighted to present our manuscript on the role of the deubiquitinase SYLT for liver homeostasis and hepatocarcinogenesis. The experimental work for this study was mainly done by Tony Obanik from my group. Generally speaking, our project is an ubiquitination and deubiquitination of proteins in the liver. Basically, ubiquitination targets proteins to degradation. However, ubiquitination is more than that. It also regulates enzymatic activity and protein-protein interactions. What is SYLT doing? As a deubiquitinase, SYLT removes ubiquitin chains from proteins involved in NF-κB signaling, AKT signaling, Wnt signaling, BCL3 signaling, and other signaling events. And by the deubiquitination of these proteins, SYLT is a negative regulator of NF-κB activation, for example, and also for the translocation of BCL3 to the nucleus. We and others have recently shown that SYLT expression is dysregulated in cancer, for example, in hepatocellular carcinoma. In the study I would like to present today, we address the question, what happens to the liver if SYLT is deleted in liver cells? With the help of Ari Weismann's group from Mainz, Germany, we established a Quilox-P-based conditional knockout of SYLT. In this model, not the whole SYLT protein is deleted, but only the exons 7, 8. And this results in the expression of a naturally occurring splice variant of SYLT, we call it short SYLT, in liver parenchymal cells. And this splicing version lacks important binding regions, for example for binding of NEMO and TREF2. However, it contains a preserved catalytic domain. And in these mice with deleted full-length SYLT expression, we could observe a spontaneous liver phenotype characterized by the proliferation of periportal biliary ductals and also by the development of liver fibrosis. At later time points, the phenotype was progressing and it ends up with a portal-portal bridging fibrosis. We could also observe inflammation in the livers of these mice. We could detect inflammatory cells, we could detect the induction of pro-inflammatory cytokines. Importantly, these mice were much more susceptible for the development of liver cancer upon triggering with the chemical carcinogen diethylnitrosamine. Interestingly, these mice did not only develop hepatocellular but also cholangiocellular carcinoma. The reason for the liver phenotype we observe in our mouse model may be increased NF-κB activity. We could indeed observe an increase of NF-κB activation in the livers of these mice. However, the exact role of NF-κB activation as well as the exact role of the splicing version of SYLT remains to be clarified. The reason why we could detect cholangiocellular beside of hepatocellular carcinoma might be explained by at least two ways. One way is the model we use, the ALPRE-based model here for the knockout of SYLT in liver parenchymal cells, is not hepatocyte specific. Thus we could also observe a deletion of full-length SYLT in cholangiocytes because this knockout also affects progenitor cells. On the other hand, it has been shown that normal hepatocytes can convert into biliary cells that may act as precursors for rapidly progressing cholangiocellular carcinoma. So in summary, we show here for the first time that SYLT is an important regulator in the liver, an important regulator for liver homeostasis, and can also contribute to liver cancer development if expression of SYLT is deleted. And this might have therapeutical implications. So the reconstitution of SYLT may be a therapeutic goal for liver diseases, And this might be achieved by different ways. For example, SILT expression is regulated by RAS-RAF-McErk signaling. It's also regulated by ubiquitination and phosphorylation. All these signaling events can be targeted for the treatment or prevention of liver diseases. Thank you very much for your interest in our research projects.